start recording. Be aware of that. And make me the like that. So we will talk today about the biological databases. And biological databases are mainly all the databases that have to do with, let me see this, how they centrate it like that. So biological databases are databases that are kept by researchers to share all the knowledge that they have acquired by doing the research that you usually see in papers. So this presentation I did 2 years ago. I did not, I did upgrade some things, but I need to upgrade again. But many of the basics of this presentation have not changed for many, many years. So I hope that you use this knowledge to take your own decisions later on when you work with a material. Mainly, most of it, I would say, sequences. Biological databases are kind of divided in 3 major types. Is the sequence databases, those were the first ones created. Then we have the genome databases, and those are containing many different types of information related to the genomes of different species. Then we have the structure databases. Now, the order in this slide are not corresponding to the historical order of the database. You will see in a moment, I will repeat it, but I say it immediately. The structures databases were first, then the sequences databases came, and the first ones that were created were for protein sequences, and then they were the ones created for nucleotide. And last, not least, the genome databases. And we will see a little about historically why things look like they do. The sequence databases are the oldest type of biological databases if you don't take in consideration the structural databases. And today are the most widely used. Every time you do an experiment and you find that there is a variant or a new mutant or changes, additions, deletions of any gene or any area that is related to the expression of a gene, this is at this level and will be found, and you will make use of these databases to find what is the name of the gene, what the gene is doing, if it's known or not, or if you have discovered something completely new. This is done by questioning and analyzing data from these sequence databases. And the sequence databases are divided in 2. Pay attention about this, but because yesterday you could also see that depending if you are working with nucleotide sequences or protein sequences, you need to choose the proper tools. Many tools work only with nucleotides, and some work only with proteins, but also you need to be very careful for which tools you're using to interrogate these databases. Otherwise, you may get very wrong results. We will see that on when we look at the EMBOSS package. It's a very simple, very powerful package. We see next time. And in there, you will do some exercises, so things will be very, very clear to what are the differences in tools and also matrix and what is known about these databases. The nucleotide repositories are mainly three. For nucleotides, one is the ENA, that is the European Nucleotide Archive. It was previously called EMBL, Nucleotide Database. Then is GeneBank, and then the DNA Database of Japan. All these three should contain, in theory, all public DNA and RNA sequences that exist. It's not really the case because nowadays people produce so much data and so fast that they have difficulties in keeping up synchronizing. But in reality, if you submit to any of them in 48 hours or 3 days, the data that you submit to one of them should be distributed to the others. Historically, the European Nucleotide Archive, which was called EMBL at the beginning, was created in Europe at the European Molecular Biology Laboratory. I pay attention to this EMBL because it will appear many times, and I hope that you will understand why. Not every time in sciences, in biological sciences, and also, of course, in that case, in bioinformatics, things are so logical. It's not that it's a logical thing, why the names and historically. Many things have to do just because they were made by a group, and the group invented or put the name of the lab to what they were doing. And then historically, this has been kept. Also, as I mentioned before, the structural databases were the first created, and then the protein databases, and last, the nucleotide databases. And why? Well, very simple, because the technologies that could make it possible to make the analysis at these three levels appeared. And when they appear, also the necessity to organize the data that was created. So in the very beginning, in the 60s, end of 60s, 70s, people could look at proteins and do X-ray crystallography and also NMR studies, and they could look at the structure of proteins. At that time, the databases of structures were printed in books, and these books were distributed to people. Because they were printed in books, some of the terminology in databases, biological databases, follow the same nomenclature as books. So, you know, when you have a new book, even if it's a thriller or a horror book, whatever it is, it's called a new release of, and then they say the name of the author. So the same is for this first release. That means the new version of the database when it's upgraded. It's called release because it follows the nomenclature of books. And the first people that got the job to make the first biological databases in different labs in the world were the librarians of those labs. So they organize things as they were used to organize books and, you know, the bibliography. So you will see many things are really like a librarian organized things. And we have not changed that so much the last years because it's very difficult to restart everything that is for years now, collecting data. But in any case, the DNA, when you have sequences, you need to know about the existence of these three places, because when you want to publish, the journals will ask you for the accession number of your sequences. Otherwise, they will not allow you to publish because it's a law. Most today funders give you money to your research, but when you get the money, you have to read the contract. In the contract nowadays, usually it says that all sequences that you produce should go to a public repository. There are people that try to do patents and all that, but that is another discussion. But in reality, most of the public funders, that means in Sweden, VR, which is the Research Council, they demand the researchers to put their data in the public repository, so other people can also make use of them. And to do that, you need accession number. And the accession number is not given by your professor or your university. It's given just by these three places. So you have to submit your data. They have progressed very much. So today you can go to any of them, depending which one you find is easier or closer to you or whatever you have projects there. You can access them and put your data, and then you will get this accession number. What is very, very important to know is accession number is not related to the name of the gene 100%. So when you do any job with any database that is biological database, and you find something very interesting, and then you want to publish after half a year or 1 year, the name of that gene may have changed because the other researchers have found that that was not the correct name. But the accession number of that sequence will not change. So it's very important that you make note of the accession number, because then you can go back to the database and retrieve the information. Because if you just write the name of the gene, you may go back and not find anything. You will get very frustrated. Your professor will get very angry. And the cake you have been preparing to celebrate your new paper will need to go to the trash. Only because this very small detail that can make a mess for you. So remember this. Accession numbers, they don't change. Names change. Information in the sequence change. Now, when you put the data in any of these three places, as I said before, they have collaboration. In this picture, you can see there that still the European Nucleotide Archive is still called EMBL Europe. EMBL is the name of the biggest molecular biology laboratory in Europe that was created very, very early, and most of the high-profile professors in molecular biology in Europe has been there once or twice in courses to improve the knowledge in molecular biology. So it has been the major hub for educating researchers in molecular biology. And that is the reason it's called European Molecular Biology Laboratory, and is located in Heidelberg in Germany. And they announced, this is just so you know for other purposes, they announced courses in molecular biology, image analysis, nowadays bioinformatics, and many, many other subjects. But when they created this laboratory and have the latest development in molecular biology, they were also involved in creating the first machines that were automatically sequencing sequences. And of course, they created and did so many sequences that they needed to organize this. Because before that, researchers had the sequences in books, in the labs. They could not share them. When I was a student myself, I had my first sequences in a floppy disk. And you may ask why it's called floppy disk. Well, I guess you've never seen one. But it was a floppy disk was this size. And it was thin like paper, but in plastic. And when you did like this, it was really floppy. And you could destroy your data by not handling this very carefully, because the thing could break, which happened to me. I learned very fast to have backups of everything. And that thing was the way that we distributed our knowledge, going to conferences and telling people, do you have some sequences you would like to share? And people would say, yes, I have some sequences. And then you establish collaboration. But that was absolutely primitive and not very efficient way of sharing data. So the reason that people realize that they have to create these very big centers, they cost millions to maintain, millions of dollars a year. Why do they do this? Well, very simple, because they did a few experiments that gave such great results that people understood why it was so important to have this. And the first one that I at least I mention usually, that I know personally, was a group that have a collection of sequences that were isolated from the brain of an ape that had tumors. And this other group that were working with tumors in humans, they were looking also for many genes, among other genes that are to do with growth factors. And what happened with these two groups, having the floppy disk and just sharing data and using one of the first bioinformatics tools that could compare one sequence to already existing sequences, was that the best match and very high match between the newly isolated growth factor PDGF-B was over 68% equal to an oncogene derived from a virus that created tumors in the brain of apes. So people immediately understood that just by searching similarity, they will understand functions and they will establish completely new areas of research. So this is just an anecdote, just to tell you that this is why the interest started. And today is something everyone does. Every time you have a new sequence or a sequence variant or something interesting, you just go to these places, use a very simple tool like BLAST, and you search in the databases. And you will always get answer, something similar. But that doesn't mean that you are done there. Then you need to understand a little more about how databases are built and how to really say this is a valid or this is very bad result anyway. Hits will also, you will always get, but if it's good or not, that is what I hope that you will learn from today. And so what are the contents of these databases? They are super simple. As I told you before, these things were created by the librarians. So the librarians usually have the library as you have your, if you still have, I don't know if you use that small books or at least in your iPhone, you have list of friends. So you have the name of the friend, you have then the address, the email, maybe, I don't know, the WhatsApp address. So you keep in different fields. If you are a Swedish citizen, the most important part is the personal number, if you can have that in your list of friends, because with that you can connect to any authority in Sweden and get information about that person. Because name, you can be Lars Jansson, and it can be 200 Lars Jansson in Sweden, but every one of them has a personal, unique personal number. So it's very similar to the databases. And as you can see, this is so easy in reality, in the way that it's organized. First, of course, you need to have a sequence. Otherwise, it's not a big idea to have a sequence database. But then you need to have the accession number. And I repeat it very many times, the unique identifier for your sequence. Then you can have references. That means if this sequence is coming from a project where it has been a publication, that should be there. Taxonomy data. That means from where did you isolate the DNA or RNA that is corresponding to this sequence. If it's protein, the same. Keywords. Keywords are, for instance, derived from brain, related to cancer, whatever. These are keywords. Many places on internet that have databases allow you to search on keywords. Sometimes you say, oh, I know this gene really is involved in this process, and I know it's coming from there. And you put the two names there, the two words, and you don't get anything. Why you don't get anything? Because keywords is one of the fields that the The tool will search in. And if you say searching keywords, and the person that put the information in the database about this exact sequence did not do the job well and just put isolated from human, but didn't put brain. And instead for just putting cancer, put disease, then you will not get the answer back, even if the sequence is there, because this is very important. The data and information that is on the nucleotide databases is put there by the people that did the sequencing. Some people are very good at their job. They work very meticulously. They send the professor to do the job that knows all the details. So fill in all the questions that the database people make. But some people maybe produce too many sequences and put several students, and maybe the students do not have the capacity or knowledge yet to fill in all the fields correctly. All that goes into the database. There is no very much control over that. So errors that people do when they're putting things in the database can be there for 20, 40, 50 years, and no one is correcting that. It's not manually controlled. So the quality of nucleotide sequences may be not that good as you think. Now, I repeat this, when you're working with nucleotides and you're working with nucleotide databases, use this knowledge. Is super wonderful and marvelous, but don't trust 100% in this. It can be errors and it can be things that are not real, that you, with your work, and if you put a lot of effort, you can correct that. You may have right, not what is in the database. Pay attention to that. Don't start inviting for cakes and to a party because you found a mutation that's causing something, because it could just be an error in what is in the database, and you got the right sequence and you can correct that. So it can be messy. Remember that. Is no manual curation in nucleotide databases. So the database formats, this is also something that nowadays is not as big problem as it used to be, because people have understood that it's a mess when people try to use all this data and make sense of it. And they go to these different databases, and all of them have different formats. And what is a format? Well, format is that the sequence should start with a dot and end with semicolon. That defines where the start and the end of a sequence is in one database. In the next database, it has to finish with 2/. And in the third database, only with 1. And that tells the programs when the sequence starts and ends. You see? And because they have decided, because this is people keeping these databases, and because they wanted, they decided to do it that way. But the programs are stupid. The program expect to be a slash or to be a semicolon, and they will say, sorry, but I don't find any sequence in your data. And that's usually is because you are using the wrong format. You need to understand this about formats, but it's not a big issue nowadays. There are many tools that convert from the format that you extract from the database to the format that the program that you will use understands. But be sure about this, because I'm not exaggerating to say that 50 to 60% of the times when people call the bioinformaticians for help because things are not working, because they downloaded data and they cannot do analysis with the program or tool that they were told to use. And it's only because they're using the wrong format. And as I told you, if you have 3 sequences, you could even transform those sequences by putting it in a text editor and changing the slash for the semicolon. But if you have 20 million, I promise you that you will never do that manually. You use a tool. And next time we meet, I will show you a tool that can convert from almost any format to any format for you, both at the command line, which allows millions of sequences at the same time immediately, or using a web interface so you can cut and paste whatever you're doing. So it's not a problem, but you should know about this issue of format. And for instance, an example here is very small, but you can see it later when I upload it. This is a so-called SwissProt format. Is very, very similar to the EMBL format. And it's just the way as it's organized. And as you can see again, it's really following the librarian's way of organizing things. It's the name of the gene, it's the accession number, it's a reference that are known. If other entries in other databases are known, they will put them there. For instance, if you find a sequence of, in this case, a protein, if you have connections to other databases that have information about the structure or about the expression or about is this run in two-dimensional gel electrophoresis, so you can look at the dots, even that will be in this format. But for many analyses at the bioinformatics simplest level, you will only need, because this is information that you need to prepare your paper or see how you're going to work with next experiment. But if you want to use only the sequence, let's say that this format is French, but your program only understands English. It's like us here talking today. I speak Swedish. I know that many of you speak, I don't know how many beautiful different languages, but to understand each other, we use English. In bioinformatics, very clever people, I would say two people, two professors, yes, did something very radical, very simple. And that was to create one format that is kind of the English in bioinformatics. And that is the so-called FASTA format. And the FASTA format is super simple. It means that the first line in the text containing a sequence should start with the symbol minus, bigger than, and have all the information that you like. You can write very long, but it's unnecessary. Usually you can write, this is my file, or this file is from experiment 43, if you want to be very accurate, band 48. You create whatever you want in the first line, because that will never be considered as sequence by any program. But all the programs are expecting that the second line starts with a nucleotide sequence or a protein sequence. Super simple. Again, you can do this manually if it's only 1, 2, 3 sequences, but I recommend that you use a tool if you have many sequences. So the English of bioinformatics, FASTA format, super simple. Now, remember, the most important part about nucleotide databases are they are built by the people that create the sequences, and is only the only reason that a lot of people put the data there is because otherwise they had to give back the money that they got to do research. It's not that everyone is so generous, and also because it's a requirement by the journals. And why is that? Well, because when they give money, the funders, if it's private or if it's public, they want that the data that you produce is also shared by other intelligent and nice researchers. So maybe they can extract more information to the data that you produce. Because nowadays we produce so much data, but we are usually interested in 1 or 2 things. And the rest of the data we don't even look at. But maybe other people are interested in that because they are looking for other areas that the data can help them to do. So for that reason, the data is provided by the creators, the authors, the researchers, but it's never checked like no one is resequencing to check that your sequence is properly done. And there are labs that produce excellent, and there are labs that want to be the first in the world, and they are so fast that they are full of errors. So all of them are in the same place, and there is no kind of quality that is higher for the good quality and lower for no, that doesn't exist. They're all equals, the sequences. And when you do a blast, the one that's coming first is the one that is closest to yours, but doesn't mean it's the best. Remember that. That's the lecture. So for protein databases, it's a little different. Protein databases were created before a little later, but they were created having sequences from before the nucleotides. And if I cannot ask you, because we're not in a classroom here, otherwise I will make a lot of questions to you, like, when did we start sequencing nucleotides? Year, which years? And why? Or not only why, but what make it possible? I can tell you the answer immediately, so you don't suffer. We researchers started sequencing in the, I would say, late 70s, and it did not start to be massive until the beginning of the 80s worldwide. And it was because Sanger, that got the Nobel Prize in chemistry because of that, created the Sanger sequencing method. And after that, everyone that could learn the technique started using it. And that was in the 80s. So for that reason, the EMBL database was created 82 or 84, I don't remember exactly. Then the necessity to create one for the proteins came up, and you can see in bioinformatics, the needs are there. Everyone is screaming about, and we need a solution. But in most cases, it's one person or two people that decide to make something to solve this issue. In this case, it's a researcher in Switzerland called Amos Bayroch that started a database called SwissProt. And as you can see, the name is very logical. It's a protein database created in Switzerland, SwissProt. So again, if you know the history, you will not forget what are the differences and why the names. Otherwise, it's very difficult to remember these things. It's so many names. But he created this together with the Swiss Institute of Bioinformatics and the European Bioinformatics Institute in the UK. The database is annotated manually, and I will repeat this several times because this is the biggest difference from the nucleotide databases that are not annotated manually in the same way. It's non-redundant. It means that if you put there one protein sequence that is coming for, let's say, the actin gene from human, when it enters, they are not going to accept a second and a third and a fourth sequence of the same. They may accept actin from rabbit, from horse, but those are different species. Even if at the level of similarity, they are 90 or 100% equal, but it's different species, so they are unique. That means non-redundant. In the nucleotide databases, they accept everything. So it could be that there are 20 competing groups working with the same question, and they all produce sequences that are related to actin. And then you may have 85 copies of the same gene because they're all coming from different groups. So when you do a blast, you may have 85, 100% hits. That doesn't mean that there are 85 different genes. It's just that the sequences are coming from 85 different groups. And if there are small differences there, it may be that one of them is coming from a patient that do have a mutation, or that it just was an error in sequencing. So you see, that's the difference between non-manually curated nucleotide database and manually curated protein database. A very, very big difference. And the database is constructed, again, in more or less the same way as I mentioned before. This means that you have there the code, you have the ID, you have the accession number, you have the date when this was uploaded, you have the description, the gene name or names, because sometimes there are discussion in different groups what the name should be. Some people want to put the name of their daughter or the girlfriend or the husband, and some other groups want to be very methodical and want the gene to be called F3/A because they're following some kind of nomenclature. Then they go together in a conference and they all agree that there will not be the name of the girlfriend and it not be the F34, but a completely new name. And in the nucleotide database, you will have the same sequence having 3 different names. So be aware of that. But the accession number is the same, only one. Again, they will put their annotations. And as you can see in the picture, in red, it shows which areas that are manually annotated. This is super high quality. This is not a student that got punished to put 400 sequences in the database and just cut and paste and did not put a lot of attention to each sequence. This is a team of people that are working to fill in all these areas. And as you can see, in every single entry or sequence in the protein database SwissProt have information, if it exists, back to the nucleotide in the EMBL or gene bank, or to other databases that are very specialized. If any of you, may I see your faces, guys, all of you, because I want to do a few questions and I only see 3 people. It's difficult to see if I'm putting any kind of new knowledge or I'm just repeating myself. So my question is, any of you that knows what rebase is? The database rebase. Any of you that is using restriction enzymes to cut DNA? Any of you have done that? No one has worked with DNA at all. Anyway, if you're going to do that in the future, when you want to use restriction enzymes, just to see, let's say that you want to build a new construct. You want to put a promoter together with a gene and a gene variant, all that, you need to have molecular biology. To know how you cut the pieces of DNA, you need to do it with the restriction enzymes. This database called rebase is updated every month, contains all the information about the restrictions enzymes that the market can sell to you. So when you are working there, usually in many labs, they do have a small machine containing a small bioinformatics program that can do, you can put the sequence and ask, please show me which restrictions enzymes can cut, and you will get a very nice table with figures and all that. But what is wrong is that most of those machines, no one has updated the rebase database for years. So you may miss the latest restriction enzyme that could have done the miracle for you to cut the piece that you needed. So you need to upgrade. That is just one example. There is another there called transfac. Anyone that knows what that is? Should be super important for you if you're going to work as breeders, because most of the genes is not that the genes are mutations that make the trick to make the plants grow faster or slower or have taste or all that. Is in many cases the areas that govern the activity of the gene. That means the gene is on, is off, is producing huge amount of the gene, or is producing very low amounts. All of that is taken care by transcription factors in front, in the middle, or in the back of the genes. And this database containing all the known transcription factors and where they bind for you is super important to know, because if you find something different in a gene, it may be that it's just one nucleotide difference between the mutant that grows very well and the one that doesn't, and it's because it's in the middle of a place where a transcription factor binds. And that information you can find in the transfac. Now, you see, you can go back to this figure because it is like a map of many, many, many databases. So you don't need to remember this now. It's impossible the first time. But later on, you will see how these things work. Let me see here. Sorry, I was in the wrong place. So now, why I'm talking about manually annotation that's different from just sending the data yourself as the producer of the data. Well, in the Swiss Prot, there is a very big group in a house in Switzerland. They receive the data from the researchers, or they can, many cases, they extract the information from a newly published paper, and they take the sequence, and that is taken care by an annotator. That means they're sitting in a room with computers, very knowledgeable scientists. Some are specialists in plants, some are specialists in animals, some in fungi, some in bacteria, etc. I mean, in the biology. They take these new sequences and they start filling in all these fields. You remember, like your telephone book, but in all the fields that are necessary. Is it known that this gene is involved in a disease? If it's involved in something normal for growth of a plant or a human or a cat, all of that. And when they do the work, it's like doing a master thesis, but shorter, about one gene or one sequence new. When they do their job, that goes to a head annotator. That means it's a person that have a higher degree, higher than postdoc, that can decide if this work was well done or not. If it's badly done, they send back and do it again. Or if it's perfect, they will say, yes, this is good. And that will go to the head of the Swiss Prot. And it's only the head of the Swiss Prot that put this new sequence with all the information in the database. It used to be Amos Bayroch, but he's now doing other things. And now is Scenarios, another researcher that took the head of this work. Now, you see what the problem is and about the differences between the nucleotide and the protein database, like this one, that is manually annotated. So when you search and you get hits, you can be much more confident and have much more data when you find in this database than in the others. But you see, the Swiss Prot looks like this. You can go to Expacy. This is one side. You will get a list of URLs and names, so you can go and visit later on. In Expacy, anything that you want to work with proteins, you will find information there. Free tools to download, install in your own computer. You can run things there. You want more information. You want even to make a small tutorial to learn how to use things. You find it there. It's the home of protein science in the world, I would say, without exaggeration. So the question is, it's beautiful, it's handmade, but it's like, you know, pieces of art. Not everyone in the planet can have pieces of art in their home because it's too expensive, it takes too long time, and we are more and more people, as you could see in my curve the first day. So how can we do? The researchers are producing more and more and more nucleotide data, and you can with bioinformatics tools, identify new genes that have never been discovered at the protein level in the sequences of nucleotides directly. So how do we do? Because as you can understand, it's not fast enough, this process. Its quality is very good. We would like every single protein sequence to be taking care of them, but it's not possible. So what happened is that the bioinformatics community opted for taking every single gene that is deposited into the nucleotide database. At the time was called EMBL DNA. Now it's called ENA, the European Nucleotide Archive, but the name for the protein part remains because historically was the name. So it's called translated EMBL. So what is translated EMBL? Is all the sequences that are in ENA that are by a computer annotated and translated and protein created by the computer. And all these new sequences that are coming into the ENA, and then after a while go into Tremble. And Tremble is a protein database. Now, it's not manually curated, and it has the same level of quality as the mother sequence that is coming from the nucleotide part. So be aware of this, because when you search with BLAST or other methods into the protein databases, you will get a lot of results. But now, after this lecture, you will see on the side of your hit if the sequence is coming from SwissProt or if it's coming from Tremble. Then you know if you invite for cake immediately or if you have to wait until you announce your new discovery, because it's a matter of quality. So things are going very fast, and the Tremble is very good database, no question about that, and you had to use it, but it's just so you know the differences and what you can trust more and less. Now, the Tremble looks more or less the same as the format of the ENA database and the SwissProt. Why? Because it's the same people involved. I can tell you that in the beginning of the 80s, middle of the 80s, a network of bioinformaticians was created in Europe. It was called the EMBnet. EMB, you just recognize European Molecular Biology Network. And that network was, the idea was to put all the bioinformaticians that were working in Europe to find standards and to do things together and share the databases and share also knowledge about these things. And that's the reason that many things look the same, because people met to agree how to do things. And I can tell you that in the beginning of, in the middle of the 80s, you could put more or less every single genius bioinformatician in the whole Europe in just a small meeting that could take 30 people. That was the world. I mean, things started then. Today, you see these people in the pictures when they met. They're all people that are super well known, and they have done all these tools. I mean, the people that did the cluster to make alignment of multiple sequences, the people that did the SwissProt, people that did the nucleotide databases, etc., etc. Very few. And as a reason, things look the same. No one was reinventing the wheel if it works. Now, what is very important also, databases are alive. They're not dead. The only one that is really kind of rigid is the ENA and GenBank, because everything that enters there almost never get out, even if they discover an error. I know of many sequences that were wrong. We know that. But those sequences were entered in the end of the 80s. They're still there. So still you can do a search and you can get a hit with that and say, whoa, this is a mutant. No, it's the same sequence that has been making problems for us for years because there is no depuration. But on the level of proteins, it's different. You can see in this schedule here, this schedule, many people don't know about it. So it's kind of exclusive for you. The life of a sequence. When you get a sequence, a new sequence, let's say that you are working with a very nice project and you isolate a sequence, you put that on ENA because you want to publish. And as you see there, where it says cDNA genomes, that will go to the EMBL or ENA. When it's there, the people taking care of the data will translate this in a computer without even asking you, and will put that into the Tremble. And the Tremble will be there with a lot of sequences that are not in the main Tremble for a while because they're checking details. Then it enters to Tremble. And when it's in Tremble, it could be that there is a group in the SwissProt that discovered that this sequence, protein sequence, is very high quality. They do the annotation. That makes that entry in Tremble disappears. But you will find it in SwissProt. Why I tell you this? Because I have had so many people that are almost crying because they are writing the paper and they know that they found this sequence in Tremble and it's gone. They say, my goodness, how can I prove that I did all this? But they should really buying cake for everyone because the sequence was such good quality that entered to SwissProt and disappear from Tremble. Because you remember, Tremble is machine-made, but when it's handmade, it's higher quality. So databases are not dead. They are living organisms in some way. So they're changing. You should know this. And this is why I repeat it. Keep the accession number. Otherwise, you will have problems finding things when you are writing things. When you are working, fine, you do a lot of things. But make notes as you do when you're in the lab. You put the temperature, you put how many volts, and you put how many milligrams you use. When you're working with a computer, you put when did you do this? Where did you do this? And what is the most important, really the most important of everything? Write down the version of the program that you're using. Because it could be that the version that is at the moment that you do analysis has a computer bug, a problem. And when you did your analysis, you get something strange. And you don't notice that because you trust the program. But then when you publish, you try to publish your paper, maybe a good reviewer will try to repeat what you did. And they will not get the same result. Why? Not because you did anything wrong, but the version of the program that you use and the reviewer is using 1 year later or half a year later is another. Maybe they solve the computer bug in that. So make always a note about the version of the program that you use, because they are all, it's like when you use, Word, or you use any program in I don't know, some of you use Mac, many of you use Windows, but they are always updating the operating system. And these can cause problems and things can look wrong. The same thing is when you make a note of the program that is behind anything that you use, you write the release version of the database that you use. So let's say that you did a BLAST to know a little more about the sequence of interest that you have in your work. Then you write the protocol, material and methods for your article. Please, please never, ever write, I search the BLAST database at NCBI. Oh, that paper, if I'm the reviewer, will go back immediately. Because BLAST is not a database. is a program that takes a sequence and search in a database. So you need to write the version of BLAST. BLAST is always upgraded, can do errors. It had happened, huge errors. You cannot believe this. And the database is also updated every month. Every month is a new release. And let's say that you did an analysis today. You did it in version 84. When the reviewer is checking is version 89. The amount of sequences in that database are 10 times more, maybe 100 more. So all the numbers be different because the statistics will change. The more sequences you have, the more hits you have, it will be different. So you need to put the versions. Is that message super clear? So I hope that I will not see a name in a paper in the future when I see these errors. It's like not writing what was the temperature in the gel concentration of agarose if you run a gel, or if you run a protein to put how much acrylamide concentration you have. The same. People need to repeat your experiment. So, and this just show how fast this is growing. It's just exploding. I mean, the databases are absolutely crazy. They are just increasing and increasing. And if you ask the question why, well, because the prices are lower and lower. And our curiosity is more and more growing because it's possible to solve problems today in ways that they were not possible a few years ago. Now, as you could see, there are many databases, many names, it's very complicated, and everyone in bioinformatics realized that this is the case. So what the communities in bioinformatics has done is to put all these databases together to one. So when you search with a protein sequence in a protein database, or you go to a BLAST, a place when you can use BLAST, you will not search to the individual databases. You can do that, but in most cases, you will search in UniProt. And UniProt is very easy. UniProt is just all these databases in one. So when you do this, and you know now that you will search against most of the known databases, and that when you get a hit, you can tell if this is high quality or lower quality, and you don't get a very good hit. Let's say that you're working with a completely new discovered gene, and there are not many other projects that are being working with plants or bacteria or fungi that are similar to yours. So you don't get a good hit, and you say, my God, this is not helping at all. And you need to design experiments to prove what the function of this gene is. Then it's not enough to do a BLAST, because the information is not there yet. It's you that want to get the Nobel Prize. So you need to discover something. Well, I would say that don't give up. There are hundreds of beautiful bioinformatics tools. Of course, they're much more complicated, demands more of you, but you can get much more information from that. Some simple ones are that you can go and search for motif and domain protein databases. What does it mean? It means only that if you think about proteins, most proteins are like Legos. They can have one area in the beginning and one area in the end that have functions. And the areas in the middle are just structural to keep these two functional areas together. To get information about the function of a new protein that you have discovered, if you do a BLAST, maybe this area that is functional in the beginning and the one in the end are very short, a few amino acids. And the area in the middle is kind of long. When you do a sequence similarity search with BLAST, that will not give you very high results because the similarity is not very high. But the similarity in these very few amino acids in the beginning and end are absolutely very high because it is functional. They need to look like that. And the distance between these two functional groups needs to be absolutely always the same. That is something that makes these proteins members of a family or representing domains or motif. There are different names for the different ways that people have taken care of this protein. And there are many databases that are being created with this. The basic way of creating these databases is very simple. They take a lot of sequences that kind of well-known function from different, if possible, from different species, and they align them in the whole length. And then they see what is the minimum common denominator for these sequences to still be active. And then they take these areas that are very well conserved and call that motif or domain. It depends of the project that started this. So there have been many projects out in the research community that have created different algorithms, different rules to when and how to call these areas motif or domains. So those are the differences. They also use different methods to search. You may remember from yesterday, you talk about HMM a little. So these are the methods that are used to search with these in these databases. But what is nice for you to make life a little more easy is that most of them, not all, but almost all of them, went together to just one database called Interpro. So nowadays, you only need to remember for sequences is ENA or GenBank. For proteins is UniProt, contains all of them. And for domains, motifs, and all of this, as you can see there, is ProCite, ProDom, Prints, PFAM, SMART, all of them are together in one called Interpro. So if you have problems finding good match to sequences that tell you more about the function of a gene, go and use the tools in search in the databases related to Interpro. Now, next time we meet, we will do a simple exercise on how to put together several sequences and construct your own motif to search in a motif database. It's super simple, super powerful if you follow that that tutorial that we're going to do, because it's the only way to get information when you are in the, as we call it, twilight zone of what is known. But it's only in the twilight zone that you will fetch things that give you the super paper. If you just are going for known things, it's not very much to win. So you need to get these extra things. You will be very happy when you do these things later on. So Interpro can be visited in that address. As you, again, I will give you the recorded, if I remember to record, I forgot. I hope that I'm doing it. Oh, it's recording. Yes. Oh. Sometimes my brain is too busy with other things. I forget them. I even forget where I am. Yeah. So this is at the EBI, and the EBI again is the European Bioinformatics Institute, is where ENA is, but they also take care of Interpro. And you can go there with a sequence and search. And what it will give is not only match to sequences, but also figures. So tell you which areas that are conserved and may have a function. As an example here. So you can see here, these are example of motifs. So you have there areas that are very well conserved, but as you can see, most of the sequence areas are not very well conserved when you compare several members of the same family. But if you have a new protein and you see that it follows the same pattern, then you can go and do this search. And when you get sequences that are in the same family, you go to SwissProt, and then you can see on the fields that are in the hit, the connections to all the other databases. That means, where is this usually expressed? What function is usually known? And then you get a very nice, you can write down a small paper saying, okay, in most members of this family, this is related to protein transport, or this is part of a receptor, or this is a cytoskeleton protein. So that has to do with the structure. Then you go in your group or whoever you're working with, and you say, we need to prove that what we found in a bioinformatics analysis is also taking place in real life. And that means you design your lab, and you design an experiment, and you do it. You're not doing it, just by guessing. You do a very qualified guess based on what is known from other knowledgeable databases. So this will, I would say, save you incredible long time. And I guess when you work with plants, you have to wait for a very long time when you're doing experiments to look at things. So just doing it many times will not help you as much as doing a profound bioinformatics analysis that tells you exactly what you could expect. And that means that you can narrow your experiments in time and also in costs. So it's super to do this. A lot of people that do this are so happy afterwards. And again, examples of these entries, if you get hits in the motifs databases, this is coming from the protein family database, PFAM, very easy, protein family. As you can see, if you just see PFAM, it doesn't tell you anything and you get confused. But when you know it's protein family database, you say, my goodness, why do they need to complicate things? Well, I can tell you that one thing that we have that is very bad in the bioinformatics community is that most bioinformaticians are also very good programmers, or people that works a lot of with computing. And in computing, most people use the hands to write command line. And when you want to write, you don't want to write very long commands. So you don't want to write, show me the long list. You just, after a while, you just write ls. That means long list. If you want to show the processes, you write ps, not the whole phrase. And that mentality of shortening things also is mirrored in the way that people put names to programs in bioinformatics and also to databases. So I apologize for my colleagues and myself, but it's because with time, you will see it's so much easier to write things and make things work in the scripts. So that's the reason things look difficult, but they're not. It's just the first time you have never seen this, so it's not so strange. We end with the protein databases in the reverse. We end with the ones that started the whole thing, and those are very easy, are the structured databases. The major one is the PDB database, contains most of the well-known sequences. Most of the the structures there, they are worked out looking at the X-ray data. So in the beginning, very beginning, they did X-ray, and that means that they could do in layers, many layers like this, and then they reconstruct that by cutting in real life the layers, and then they put the layers in real life. So in the past, you could look at a three-dimensional structure of a protein by just walking around the table where they have reconstructed this. Those are the first ones that I saw when I started at university. But in the 80s, they already started using computers to reconstruct the proteins. So today you will see most of these very complex, like in the picture, in there. What you need to know is that the PDB database that contains all the information of the known proteins, that is far from what we need to know in the future, is really a need of have millions of structures. We only have thousands to understand how they dock, why they don't dock when they, a mutation makes that the finger is gone. So that means no grip. But when you have all fingers, it's a firm grip. The same is with proteins. They need to have the structures. They are not saved as pictures, as the picture you see there. The information is saved as text. So when you go to the PDB database and you, let's say, you download the structure of a protein of interest, you will download a text file. To see the structure, you need a program that reads the instructions in the text. That means which amino acid that have this property or that property, or when it has to do a coil or a beta sheet, and that will be drawn as a figure, as you see in the picture, by a program. Most of these programs are free. You can download several of them in Expasy, as I told before, the place that have all the tools for proteins. And there are some commercials that are incredible. So there are tools that you can dock several structures together, you can modify them. You will see at the end of this course, you will get with you a free tool that you can even distribute to your friends and colleagues, that even there is an inbuilt viewer for three-dimensional structures that are also, when you modify an amino acid at the sequence level, you can see where in the structure that amino acid is located. Very nice. So all of that you will get later. Now, what things are being changing is, like the last 2 years is an explosion in this area that has been kind of very difficult to work with, because you need to have crystals to have the three-dimensional structure, and it's very difficult to produce crystals of proteins. But now there is a new program, artificial intelligence based, that is called the AlphaFold, that is revolutionizing the area of protein structures. So there are thousands and thousands of new structures created just in the last few years, and this will increase even more. Many of them may be wrong and maybe exaggerate the accuracy of the program, but this is showing the future. Most of the things will be done based with artificial intelligence, and this will accelerate in ways that we could not even dream about only 3 years ago. So before, in the beginning of my talk, I said we could not dream about this 20 years ago. Nowadays, things are getting so fast that I say we could not dream about this 1 year ago, because that is how things are moving. So the PDB, can you see there, is very old. It's the Brookhaven National Laboratories, and was created in 1971. And it contains all the three-dimensional structures. And I hope that this will increase faster, because you will see, if you want to see the structures, most proteins do not have structures yet. To look at this, you need one of the tools that I mentioned. Here are some names: PyMol, RasMol, Swiss PDB Viewer, and many others. There are many others. I'm not married with any of them. I'm not recommending one over the other. I'm just mentioning some of them. And they sometimes are free for academic, sometimes are commercial. That are commercial or free is not always reflecting the quality. Some free are better than some commercials, and vice versa. So select the one that works for you. And this is how the PDB files look like when you download them from from the database. It's a text file, and then using, for instance, in this case, PDV viewer, you could see the three-dimensional structure. You can rotate, and you can also change if you want to see in the way that I'm showing you, or if you wanted to see like a cartoon, as in the first picture related to structural data. So here I just will mention major data providers. All of these addresses and places are in one page in the course pages containing many databases. If you discover any of them that are dead, please tell me, because this is also something that is always changing, and I don't have all the time in the world to keep everything properly. It goes too fast. But these guys that I'm putting in these presentations, they are around for very long, and they will be for some more. Nevertheless, and this is something I want to tell you, every time you do research, and if you're going to do real heavy, nice, super duper research and produce papers, always write somewhere, cost nothing, acknowledgement. Put there, thanks the Expacy because they provided me with a free tool or something like that. Why I'm saying this is because these guys are surviving because they get money from the different countries, and they always get evaluated every 5 years. And the only way to show that they are for the good and people is using it is if people write acknowledgements in the papers. So do it, because otherwise, for instance, Expacy almost lost all the money. It was now a long time ago, it was 15 years ago, because the Swiss government was thinking that this is not very facility. They didn't understand why this should be there. And Swiss, because, paying for this when the whole world was using it. And they almost close it. And then the people from Expacy says, okay, then we move Swiss Prot to America, to NCBI. And then suddenly the politicians are funny. They say, my God, we cannot give away a Swiss product to the Americans. And only then they promise support for the Swiss Prot and Expacy again. But it was this close that it moved to America, just because they didn't see the usage, and then because they saw that this was a prestige thing. So I'm telling you, I'm not saying for Expacy only, I'm saying if you use ENA, if you use GeneBank, even GeneBank, NCBI has huge problems in getting more money these times, because many politicians don't see the things we see, and they don't think it's worth to put so much money in these things. So continue writing, and it costs nothing to acknowledge other people that are helping us. It's very good attitude. I hope you agree with me. So NCBI, of course, cannot be forgotten. They do an excellent work. The people there is people. Huh? Another thing I want to tell you. I have met the people from NCBI. I have met the people from EBI in different courses like this that we have given together. And they're super, super nice. And also they know all the problems and they are helpful. The only problem is that they receive 3,000 emails a day from people that want help. They cannot answer to everyone, but they try to put in information in the pages to help people. And they're all aware about things that are working or not. And again, I can mention as an example, so you are aware of this, why I insist that you make notes about the version of the program and release of the database. 20 years ago, you could do a blast search in the morning and you got certain results. And then you did a search in the afternoon and you got completely different results. Why? Because the structure of the BLAST databases were not made for so many entries, so many sequences. They never built it for millions and millions of sequences. So what happened is that the indexing of the database was wrong. It was it was not prepared for this. So they found a solution. Now it's not a problem. But that year, they had problems all the time. Every time they upgraded, they had to fix things. So researchers got really in trouble and mad because they didn't understand why they got so different results. But it has to do that it's people working with this, finding solutions, and what we are doing in many cases has never ever been done before. So of course you do. It's like experimenting. No one has built a database of nucleotide, of petabytes of data. Most people don't even understand how much a petabyte is and how many hard drives that you need to keep all this alive. It's just never been done before, and the first times you do it, it may be errors. So again, make note, because you may be super unlucky that day someone did a mistake in the computer, and these servers are kept by people. If you know some Unix, I can tell you that some things you never do. But sometimes when it's Friday afternoon and you are so tired after the whole week and you're just looking to go home, you may write a command that erases most of the files you want to erase. And when you say return, you discover that you are not in the place that you thought that you were, but you were higher up in the hierarchy, and you erase the whole hard drive in your computer. And if you say you're putting my legs, no, it has happened to people I know, and you know yeah. At least most people that are in this level have backups, but to recover a backup, it takes sometimes days when you have this big system. So I'm just telling you, make notes of what you're doing. Things happen. And then the EBI, which is the one in Hingston, outside Cambridge, where they have a lot of facilities. They have a lot of databases, and they have a lot of labs. They are really leading in hundreds of worldwide projects. Now, I will end here. I know that has been very long, but I take it in one row because I'm recording. Otherwise, it will be a mess for you to see what it was, what. I will end with the genomic databases. And genomic databases, as you understand, also from my previous talk, they were only possible when the sequencing technologies just were mature to create and do this in a much more affordable way and fast way. What is difference between the protein and the nucleotide databases is that the genomic databases are more like a Swiss Army knife of information. And also, as every single genomic database is related to just one species, it's so much easy to find proper and fast information than just going to NCBI or to ENA with a sequence and blast, because you will get hits with hundreds of other species unrelated to what you're looking at, and you will have a mess. When you want to have something from one species and you know which species you're working with, go to a genomic database, because then you will find accurate and much more fast. Now, I'm not saying not use NCBI and EBI nucleotide databases. No, I'm saying use them when you need to search for super unknown. I mean, you're looking for things that you want to fetch. It's like in the ocean, fishing. But if you know that you need to cook for tonight, it's much easier to go to the market and buy a fish that you buy in the market already done. Genomic databases are like that. Everything is organized. You put your sequence, you have a hit, immediately, graphically, you will see the protein that is corresponding to that, the exons, introns, promoter regions of that gene. You don't see that in the nucleotide or the protein databases. So start always your search with a genomic database, if it's available. I don't know which organisms you're working with. Some of them do not have these facilities, unfortunately. And if you ask me the question why, it's very simple. There are rules. So for instance, the cassava genome is not in the genomic browser of Ensemble yet, because they have the following rule. They put all these systems that I will show you in a second in place only if the group, the project, or the consortium that's working with the genome of a species put the DNA assemble of the genome to NCBI, but also the RNA data. If both of these things are in NCBI, they will put it into the genomic data, because the people that are taking care of genome browsers, they don't produce the data. They just make all the data accessible from one point. So there are many species where the project leaders or the people keeping the project, they try to keep it secret, they try to keep it for themselves for so long that most people create own solutions and you don't find them in the public repositories. It's just very, very, very based on politics and people, not so much about the system. So there are several of them. The major one that I mention usually, and even more when it's plants, because it's the one that really care of plants, is Ensemble. Ensemble has most of the mammalians, most of the bacteria, most of the fungi, and most of the plant genomes that are being produced. The USCC genome browser is more dedicated to animals, mammalian and human. NCBI is more like Ensemble, but NCBI has excellent tools in everything, but they are not very good in when it has to do genome browser. You can visit all of them. The addresses are there. You can judge by yourself what you like most. What is nice between these guys is that they also collaborate. So whatever you find in one genome browser, just by having the coordinates, as you have, you know, when you have maps, you know the coordinates in a map, you can go to another map and find the same position. The same here. If you are in Ensemble, you look at the coordinates, and in the top, even many times it says, go to USCC genome browser. You just click and end in the same position, in the same gene, and the same nucleotide. It's beautiful. So the genome browsers, the one that you're going to do an exercise later on by yourself, you have 3 days to do this, is Ensemble. And the reason I chose this for you is because it's the one that have not only mammalians and concentrated in humans, but also have plants. Now, there are many plant projects that have their own genome browsers. And there is another project that allows you to take any type of assembly data and put your own data if you have a bioinformatician in your group. So there are many resources, but this is because it's one, the data is there, you don't need to be expert to use it. That's the reason I chose for the tutorial. And the genome browsers, as you see here, in this case is looking like this. You have there usually the organization in chromosomes. You have data, what are the species that are closest to this. You have connections to many databases that are connected. So as I told you, this is like an Swiss Army knife of databases, contains everything. And if you find any region there, all these databases will be connected there by graphically, but it's not just graphics. Every time you want to see the sequence, that means the nucleotide, A, T, G, C, G, G, you just zoom in to the level of looking at the 1 or 2 amino acids. And as they work as a map, that means that you can put chromosome 14, position 3,344, 2, position 3,582. And that region will be shown to you in a window. And then you can zoom in and out. You can scroll to see which other genes or regions that are or to the left, to the right of your sequence. As sequences in a genome are, as you know, they are from 5' to 3', but also they can code genes in the other direction. You will see everything in both directions. So there are genes in both areas. The more annotated a genome is, the more data it will be there. So if you look at the human genome, that is the best annotated because it's where most of the money is. You will see hundreds of entries from databases, and you will see mutants. You will see where the restriction enzymes are. You will see where the transcription factors bind from many, many projects. Then you go to other species like the cow. The information is much less because the people working with the cow, they don't provide as much information to these guys as the human does. The same is for plants. Some plants are super duper well annotated with a lot of data, and some plants are very little. But all that is depending on the community. So it's community work. The more the community put there, the more is there. And again, it's people working in these places. You can write to them. And you can even tell them if you discover an error or you discover something beautiful you want to share, you can write to them. If they find that this is correct, you will be so happy to see that in the next release of this genome, maybe your comments and annotations are up and visible. They do that. We have even done that in courses. So you can see here, you can look at the data gene by gene. And then you can see there the organization of chromosomes and of introns and exons. And you can modify the way you see the pages. That means that what you see when you get there is the default page. But these pages, as the one you're seeing at this moment, they're not static. They're not just waiting for you there when you click. They are made when you ask to see this area. I hope I make clear that dynamically produce. When you say, please show me chromosome 17 in dog from position 1 million to position 2 million, the system will go and interrogate the data in the database and will make this figure for you. And it will do it in a way that is default. That means what people usually want to see. But on the left side, you have a complete system that you can modify. You can click there and say, oh, I would like also to see the domain motif that are related to the different exons in this gene, if it's known. And if there is data that shows how the promoter is located and if there are any transcription factors there, you can tell the system, please show me that. And then you say, reload, and the page will be remade for you with much more data. So these things are dynamic. Now, as you can see, for the plants, it looks like this. The main place at Ensemble has most of the mammalians and also have some species that are there, not because it's logic, but because it's historically there. Because when they did the first Ensemble genome browser, it was made for the human project. But at that time, the other projects did the Drosophila, the yeast, the fugu fish, and a few more C. elegans. And then those are so-called reference genomes. I mean, they are for knowledge that have been acquired by different projects, because these are model organisms for research and are there to compare with the mammalians, because you can get information about which genes that are involved in the development of neural crest, for instance, or the genes that are involved in creating the eye. And then you can go back in evolution and see when these genes were coming in the Drosophila, or you can connect the knowledge to the human or a cow, whatever you're studying. So they're there for as model organisms, not for other purposes. For plants, it's different because plants are completely different. So in plants, there is another type of selection of which genomes are there. And the first genomes that went there is because they were the first ones that were public. The rice, for instance, was one of the first, but that was not public until very late. So it was not one of the first to come up to ensemble plants, but other did. And as you know, they will be upgrading, but the request is that there is not only the assembly of the genome in the public repository, but also RNA data and expression data and some more data. Then they will make this for us all in the community. And as you can see, it's rather complete. So please visit it. You have all the address. You have all the information in the course pages. Make it a visit. Now, I will end, and I apologize for being so long. I hope that you are okay. I told you from the beginning, this is my most boring talk, but it's absolutely necessary because it is the basic to understand these things. Some advices. When you work with these tools that I've shown you, a few, I mean, this is a never-ending story. Don't trust the results of only one place because they're built by people. The databases are connected in different ways. You might get different results. Now, it's not because I'm egocentric. It's because I know the real results I should get. I use my own name to search in NCBI and ask, give me everything you know about work that I have done. And you can repeat this if you want. When you go to NCBI to the first page, and there is a small place that you can ask, and you can ask all the databases. And as you can see, I get in protein sequences, 23,000, and I get in nucleotide sequences that are in GeneBank, 7,000. Okay? And you say, okay, that's it. And then there is a little more about my articles and all that. I did this a few years ago, so I have a little more, I have to say. So you don't think I have few. And then you can go to EBR, which is the other major biological database provider. And you can see there that in proteins, I have less. But in nucleotides, I have many times more. So how it happened? Well, I can explain another day. The reason is nothing is wrong. But I just can tell you that they work a little different. The results can look different. So my advice to all of you is when you are doing a work, because there are many ways of doing it, try all of them, at least if you have time, because then you can see if both have more or less the same answers. Yeah, you can be very sure that things are okay. But if you get contradictory answers or wrong, then you have to start looking at things before you run away and start writing your papers. That's a mistake that many people do. Small results, but use several. And the same is for even for using BLAST and using other tools. As I told you before, BLAST is just a tool. So you can use BLAST even in your own computer with your own data, that you can produce your BLAST databases. So let's say that you have a project and the data that you have is not in other places. You can build your own BLAST database in your computer, and you can search with new sequences in your own database. And if you look at me and say, wow, that's super advanced, I will never do that. Well, I hope that you will be able to do that the last day with the tool that you will get for free, because there is inbuilt a very simple tool there that you say, I want to build a BLAST database, and the tool just asks you, where do you have the sequences? And you show the folder and you say, go, and it will create a database, and then you can search with the same tool with a sequence against that. You will see. So never be 100% sure about things you get from any database. Use them as help to make decisions on how to perform your experiments, because it's only in real life, in the lab, that you prove that what you have done in bioinformatics analysis is correct or not. Before that, it's only indications. Usually are very good indications, but it's best to prove in the lab. So those are my last recommendations. Use several platforms. Annotate accession number. You do not trust similar or hypothetical annotations. Trust your own data. And good luck with any work that you're going to do in the future. That was for today. And I hope that you will go now to the actual pages. There are two things you will do there. One is to answer a lot of questions. Please work hard, because it's the only time you will realize I did not understand that, or I didn't hear what he was saying. So you go back and see what is this related to. And it's not because I'm testing you. It's not a test. It's just for you. It's you that will learn or not. Do it properly, and you will get answers on Friday, and then you will see how far away you were in your work, or how close. And the other thing I recommend that you do is to look at the video that I put there from other people from Ensemble that tells more details about Ensemble. We didn't have time today, but it's good to have it for you. And then to do the tutorial in Ensemble is also very useful. Everything is there. Otherwise, we will see each other next week. And next week, it will be this manipulating sequences at the sequence level. Very basic, but you will see most of the tools you see on internet behind the graphical interface are all these tools in the server doing the job for you. So it's good that you know what the limitations are and what you can trust and not trust. So I